Bismillahirrahmanirrahim. Thank you very much for handing over this session to me. And my talk is about water quality and environment, which is relevant to this session. And first of all, I will give just a brief overview of water sector challenges of Pakistan. And as we know that Pakistan is having more glaciers than any country of the world in the Indus River Basin, and Pakistan is at risk of acute water scarcity despite having with such a amount of glaciers, and its surface and groundwater sources are both increasingly stressed, and severe drought conditions persist in certain parts of the country, as it is we have seen in several decades in the third desert of Sindh, where there are issues of food insecurity and malnutrition due to deteriorated water quantity as well as water quality. And then there are sanitation issues, and the malnutrition problems with and child stunting and the deaths of the children are very common. Similarly, we have the agriculture sector, which uses flood methods of irrigation to grow water-intensive crop, which itself uses lots of water. And likewise, our water infrastructure, which includes not only the irrigation water infrastructure, but also our water supply infrastructure, is quite outdated and in poor condition. And it wastes even more water, while we have a very little storage capacity than several developed countries of the world, as we just have the 30 days storage capacity, and which is also lacking with the sediment and the silting, sediment built and the silting processes. And then we are also having problems with the intensive use of water. We are the fourth highest water user in the world, and all our water, either it is irrigation water or it is domestic water, which is used for drinking and other purposes, it is free. There is no pricing mechanism, and then we do not have much ability to store rainwater. We just capture 20% of the rain, while the rest of the world, which is in certain countries, certain developed world countries, are saving about 98% water. And if we see our water shortage trend, then we come to know that we are moving from a water stress condition to more to the water scarcity condition. And our we are also crossing declining below the water scarcity threshold of 1,000 cubic meter per capita per year. And UN estimates that this growing trend is at the annual rate of 10%. So we are also having one major challenge of groundwater declining. Our water table is declining on an average from 1 to 1.5 meters per year. And Punjab is the highest one because of its the highest population of the tube wells, which are very much unregulated. We are just developing the wells and tube wells and hand pumps without any regulation of the groundwater. Then with these very grave water quantity issues, our water quality is, directly proportional with the water quantity. And the major issue across the country, wherever you go in any province, we see the untreated disposal of municipal, industrial, hospital waste. And specifically, as we are more concerned with our human health, and then with our environment, and rivers, lakes, streams, and our, the surface water resources are the beauty and one of the biggest water resource in the country, and we are just polluting these resources, and then which is also directly impacting our environment. Then our wastewater treatment facilities are inadequate, drinking water treatment facilities are inadequate, and then again, our water supply infrastructure is outdated. And we just have adopted an approach that after every deterioration of the infrastructure, we try to build another type of the similar type of the infrastructure or the decentralized solution. And this build, neglect, and rebuild approach has actually caused the problem of lack of sustainability and the ownership of that infrastructure. Then we are also using the uncontrolled, uncontrolled usage of the different types of the pesticides and antibiotics and a different type of the restricted chemicals, and this is also causing a big challenge for our human health and aquatic life. And then there is a seawater intrusion problem specifically around the coastal region, and this is also deteriorating the quality of our fresh groundwater. And we do have these issues because of the lack of compliance to the national drinking water standards, bottled water quality standards, national environmental quality standards. And finally, we also, these are our internal water insecurities, but we do have the external water insecurities in the form of our transboundary pollution issues with India. And, we have the shared water resources with China and Afghanistan as well, but we still need to monitor how much pollution is coming from other countries. So finally, with all these issues, we are further having a risk from the impacts of the climate change, rapid urbanization, population growth. These effects are amplifying our water quality and quantity risk. And I'll just give an overview of what PCRWR has done and what type of the knowledge it has generated. And this knowledge is very effectively, it has been very effectively used by our different government and implementing agencies to address these issues and to take different initiatives to address these issues. And since 2001 to 2020, we have taken the different water quality monitoring studies, which have very well indicated that our access to safe water is is below the required standards. And you can just see that drinking water access is only safe drinking water access is 18% in rural areas, and in urban areas it is 30%. It is around 30%. And our recent water quality monitoring in the 2020 is completed, and its report is ready to be published shortly, and that will help us to understand that how this trend has now changed. So there have been different monitoring programs which have been undertaken from time to time with the different sets of objectives, and these are just a glimpse of those programs. And this has generated a big data set from the different regions of the country. And you can see this trend from the 2002 to 2015. There is a very little improvement, and just from the 15%, we have just reached to 30%. So this space of improving the safe water is not very satisfactory, and I don't think so that we shall be able to attain the improved access to safe water in accordance with the SDGs, and we need to increase our pace accordingly. And if we see the province-wise situation, Gilgit-Baltistan, where mostly surface water is available for the water supply, is having the higher risk of microbial contamination, and then Balochistan, same, these were having the very high risk in the last monitoring study. Then if we see the rural water quality, similar situation, it is again in the Balochistan, and sometimes the lack of awareness or the lack of concern of the public towards the quality of water also increases this percentage. And what were the major contaminants which are the cause of concern for us and which are very important from the perspective of the human health are microbial contamination, arsenic, nitrate, and fluoride. And obviously, the microbial comes from the mostly from the human, activities and untreated disposal of the water and wastewater. And the rest of the three can be geological or man-made, but most of these are geological concerns, and these need to be regulated in the water supply to ensure the human health and the improved access to safe water. Then arsenic is the second major contaminant, and I can see in the program that many participants, many presenters have got their research on the arsenic assessment, arsenic in the irrigation, and arsenic in the treatment. So we we have available the big data sets by undertaking a very detailed survey of Sindh and Punjab, as well as in the other areas of the country, but these both provinces are most affected, and there is a big need to undertake such type of the research which can address the arsenic contamination in a very natural way, rather than bringing the different types of the chemical or the artificial solutions. And this is just a glimpse of the nitrate contamination in the different water bodies, and similarly the different cities are here showing the status of the fluoride contamination. Then PCRWR has also undertaken a big water quality assessment survey of the water supply schemes in the country. And it was very, sad that about 37% of existing schemes in 2014 were not functional. And again, the reason of that dysfunctionality is build, neglect, and rebuild. And then there were several other types of the issues, like the lack of technical capacity and outdated there was no plans to bring, for the regular operation and maintenance of these schemes. And as a result of this, the functional schemes were providing the very, unsatisfactory level of safe water, which you can see it was in 23% in the urban and just 14% in the rural. And then rather than bringing our tap water to the safe water supply status, and rather than rehabilitating these schemes, we launched the different type of the programs in the country. The different governments in the different regime just launched those programs, and those were again decentralized solutions, which included the filtration plants. And and then after running those plants just for a few months or the few years, they just have no plan for the regular maintenance and the troubleshooting and the timely replacement of the different components, and there was no regular monitoring. So finally there was no source protection, and several factors contributed to bring those filtration plants into a very unoperational or in such a status in which they were just becoming the factories for the incubation of the bacteria, and they became the factories of the bacteria to add the more impurities in the water. Though it was the responsibility of the provincial government after 18th amendment, but unfortunately the lack of sustainability and ownership was a big issue. And after highlighting their new developments, they most of the time the local government, provincial government, and it is a trend in the country to build, neglect, and rebuild. And this has given a big vacuum to a new player, which was the bottled water industry. And they, since 2001, PCRWR is regularly monitoring the quality of the bottled water. And from 2005, we are doing it on the quarterly basis on the directive of cabinet. And you can see the trend. You can see the trend that we are we just collect the samples from the different markets of the country, and then we have the standard protocols by which we test these water, and then we publicize these results, and we share all the reports with the electronic and print media and with the provincial government. And as a result of this, there has been certain improvement. You can see this, that in 2005 its ratio was about 50%, and now there are just 10 or 11 brands which time to time come under the unsafe category due to microbial contamination or some chemical contaminants. So the PSQCA, Pakistan Standard Quality Control Authority, is the regulatory body, but still it requires a lot of improvement and a lot of check on this industry. Then we have monitored the emerging contaminants and in the surface water body from the upper Indus to the up till Arabian Sea, where the Indus meets the Arabian Sea. And it has also given us an overview of the different types of the contamination levels. And also the data is useful for tracking the different contamination sources. And the rivers which were flowing through the plains are more contaminated compared to the rivers in the upper Indus. So, and the Sutlej, Ravi, you can say Jhelum were the highly contaminated due to the existence of the different restricted chemicals in the water bodies, in the sediments, and in their aquatic life. The wastewater capability, it's only 8% in the country so far, and there is a huge need of wastewater recycling interventions, natural solutions, and technological strategies to recycle this. This will not only decrease the water pollution issues, surface water pollution issues, as well as it will increase the it will overcome the water shortage. And further, the climate change patterns, which are very different and very unique, and they are getting very diverse in the different areas of the country. We are witness of the more pronounced decrease by 0.1 millimeter decline per year in northern monsoon belt. And there are certain other types of the factors which are disturbing the rainfall patterns. And one is the global warming, and then there is an increased glacier melt, and how this is changing the monsoon pattern. And all these patterns are actually disturbing our, quality of water, quantity of water, our different, causing the floods and droughts. And so all these factors are having the big implications physically, financially, and health-wise. And one is the recharge of the water is by the contaminated water. By if the surface water is getting more pollution, it will cause the recharging to some extent more contaminated with the different types of the physicochemical or chemical or the microbiological contaminants. And this will cause not only the river quality will be degraded, but the groundwater quality will also be degraded. So there are certain outbreaks like pneumonia, and then we have the problem of the antimicrobial resistance due to the antibiotics residues, and then vector-based diseases like dengue and diarrhea. These are increasing. These have been, increased specifically during the summer, when there is a rise of temperature and the environment is more conducive for the growth of such microorganisms. And then all these factors not only are risk for the human health, but these are also problematic for our aquatic life. Like the fish, it absorbs the different types of the metals and restricted chemicals, and when humans consume such fish, it also causes the different types of birth defects and the abnormalities, specifically in the children. And these are certain glimpses of the fluoride-related dental and skeletal fluorosis, and arsenic-related hyperkeratosis and hyperpigmentation, and nitrate-related blue baby syndrome type of the effects caused by the higher levels of the certain contaminants. Then if we come to know the financial implications, then about 300 million rupees we are spending per day on health cost and the loss of income, whereas about 55 to 80 million rupees are annually spent on the different diarrheal diseases. And currently our spending is on the water and sanitation and hygiene improvement is only 1% of the GDP. Whereas by one of the estimate by the WHO, Pakistan needs about 4.7 trillion rupees by 2030 for the safely managed water, sanitation, and hygiene. Then there are certain capacity and the governance issues. We do not have the service delivery concept. We do not have the existing cost recovery models in the water and sanitation and irrigation sector. And similarly, our lack of institutional and technical capacity of the service providers. And then there are several other lack of awareness in the public, and we also import a big, set of water sector technologies, which may be quite expensive for our economy. And we have not built any capacity for developing our own water treatment products, except the few, but we require the certain types of the capacities and the setups, and specifically the advanced R&D setup to build produce our own membrane-based technologies, and likewise, similarly, the other technologies which are used for the wastewater recycling, specifically in the industrial level. So after all these challenges, we see that no doubt our academic partners and the academic institutions in Pakistan are taking a very dynamic research, and they are spending the huge resources on their research, and the students are very capable. But I think there is a big need to set the directions of the research. And as PCRWR is very much experienced, and we work on the we undertake our research on to address the country's problem, and we have to address the our problems of the country by demonstrating different technologies, and by undertaking that research which can be translated and which can be produced in the form of a practical action. So I have certain ideas and certain suggestions for the students and for their supervisors, that they should plan and undertake applied and scalable research by transforming laboratory and field R&D into actions, and those actions which can solve the problems of the country at the public level and at the industrial and agricultural level. So then we require to develop the problems driven research. Like, we need to see what challenges Pakistan is facing. Like I mentioned, that we are having the water scarcity and the shortage and the water pollution problems. So we need to undertake our research according to these problems. And one of the best guiding document is our national water policy. And we can also see the common things which are in the environmental, sanitation, and the climate change policies and SDGs. So the priorities set in these documents will be very helpful to define or to develop some research proposals for the academic research. And then I can have certain ideas, like we need the water efficient infrastructure, and there is a concept of the water smart cities. And the water smart cities are very it's a very new idea, and the certain developed countries are working on these ideas. And I know in UK and in Canada and in Netherlands, even the consortium of the universities are undertaking the water smart cities or the sustainable cities project, and how the cities are then progressing. And this is what is required by our, at least in our major cities, where, like I know in Lahore there are several good universities which can also undertake a whole city as a case study to develop or to convert into the sustainable city or the water smart city. Then we need the energy efficient water treatment processes, and we do need the climate resilient water and environmental technologies. And then the multidisciplinary approaches by including the water ecology, agriculture, health, safety, and urban plans can be nexused to come out with a practical approach and the problem solving or the problem driven research undertaking. And then finally, the application of ICT techniques and bringing industry on board, so that our research can strengthen the industry. So all of these types of the are just ideas are just to give a concept, what type of the research we are requiring, what are the challenges, and how to solve that. And no doubt the universities are doing very good research, but we need those research outcomes to be translated into actionable strategies or outcomes. Thank you very much. Okay, now we start the session, and first of all, I congratulate all the presenters for their articles acceptance in the water conference. And so let's start the session now. Before I invite the first presenter, please note that every presenter will have only 10 minutes and everyone has to stick to the time. So if there is any question, we can take the one or two questions at the end of every presentation. So we start with the first presenter, and I invite Ms. Irshad Bibi to present her research on arsenic contaminated irrigation impact on growth and uptake of arsenic in different rice genotypes. Mr. Junaid Ahmed Kori.
Yes, ma'am, I'm here.
Okay, yes, please come with your presentation.
Okay, thank you, ma'am. So, Bismillah Rahman Rahim. I am Junaid Ahmed Kori. I'm working as a research associate at US Pakistan Center for Advanced Studies in Water at Mehran University of Engineering and Technology, Jamshoro. So, this title of my presentation is actually a published research article which is published in the Environmental Science and Pollution Research in International Journal. The title of my presentation is Metagenomic Analysis of the Drinking Water Samples Collected from Treatment Plants of Hyderabad City and Mehran University Employees Cooperative Housing Society. So first of all, introduction, water is the basic need of life. So for the safety of public health, water must need quality. Its quality should be of the standard type so that humans can consume safely. So water in the drinking water distribution system can get contaminated with various bacteria which are pathogenic in nature, and this contamination may come from household, agriculture, hospitals and industries and etc. Because they directly release their waste into the drinking bodies, or somehow directly or indirectly this contamination comes into the water or water bodies, and resulting the pollutant waters get contaminated. It results when a contamination comes into water, which results various waterborne diseases including typhoid, cholera, and other diseases, hepatitis A, for example. And this is very concerned that we should take care of it. And globally, there are many waterborne diseases, I mean, there are many cases which are reported as waterborne diseases. For example, 1.3 million deaths are in all aged individuals are caused by the waterborne diseases, and about 361,000 deaths in children less than 5 years in developing countries are reported, and about 71 million dailies are only contributed by the diarrhea, and typhoid causes 20 million illnesses worldwide. In Pakistan, the condition is very worse. For example, per capita availability of the water is not only in decreasing, but the water quality is also being compromised. In a review article which was published in 2010, reported they have analyzed 7,000 samples which were pre-reported. They have found that the 71% sample contained the coliform and 58% sample contained the fecal contamination. It was leading to the 20 to 40% of burden of all diseases are only contributed by drinking water contamination, and which which causes the loss of 25 to 58 billion rupees annually. So it is in the other study which was carried by the Dowd et al. They have reported that 80% population is consuming the unsafe drinking water, and which is causing the 33% of all deaths across the country. So this graph is showing actually the per capita availability of the water in the country, which is decreasing day by day as we are misusing water somehow or contaminating water, and water scarcity. So knowing that, we actually designed a preliminary study to assess the drinking water quality in Hyderabad City and Mehran University, and to analyze their bacterial composition through advanced molecular biology techniques, and to predict their functional capabilities of observed bacteria, which I will show in next slides, that what they are capable of. So we have selected 3 treatment plants, which are 2 are from Hyderabad City, Halanaka treatment plant and Vasa treatment plant. They both are connected with the Fuleli Canal. The largest one is the Vasa treatment plant, which has a capacity of 40 million gallons per day. It runs on 40 million gallons per day capacity, and both are connected to the Fuleli. And Mehran University treatment plant is connected with Kalri Bagar feeder, and it has a capacity of 1 million gallons per day. So we have collected 12 samples from in the sterile bottles from the treatment plants and their distribution networks, and as per EPA guidelines, and one biofilm sample was also collected from the Mehran University treatment plant raw water tank. So physico-chemical parameters were analyzed of the drinking water sample. For example, TDS, these are the simple parameters: TDS, pH, EC, temperature, dissolved oxygen, free chlorine, nitrite, and other parameters, for example, fluoride, sulfate, phosphate, and other parameters were also, turbidity were also analyzed. The DNA extraction was also carried out within 24 hours of the collection of the sample. So these were the parameters which we have analyzed, and this table actually shows different parameters and techniques which were used for uh, these analysis of these parameters and standard methods. So DNA extraction was carried out from the water samples and biofilm samples. From water, 500 ml water was filtered through 0.545 micron cellular acetate filters, graded filters, and these filters were further processed for the DNA extraction through CTAB method, and DNA concentrations after extraction were carried out using qubit double standard DNA high sensitivity kits and in qubit 4 fluorometer. After that, after quality assessment of the DNA, the amplicon libraries were prepared, and for the taxonomic analysis of the bacteria V3, V4 region of hypervariable region of 16S gene was assessed with through by using a specific primer to that region, and the library preparation was carried out as per descriptions of 16S metagenomics library preparation guidelines manual of the Illumina incorporation. So the first PCR, which was simple amplicon PCR, amplicon generation, this is basically the PCR reaction mixture which we have made, and in the next, in the below table is for the PCR conditions which we have given for the PCR. After that, the PCR cleanup was carried out of these amplicons, and again the next PCR was the index PCR, in which different indexes are added to the amplicons in order to multiplex several samples. After indexing PCR, its cleanup, the quality was assessed, and in equimolar concentrations, all samples were normalized and pooled in single tube, and the samples were sequenced in MySeq system of Illumina Corporation using V3 chemistry. So after sequencing, the bioinformatic analysis of the raw reads was carried out as the quality of the reads was assessed by FastQC software, and further quality filtration and code matrices analysis and bacterial taxonomy was analyzed in QIIME2 software and QIIME2 environment, and functional predictions were made through PyCrust software. So this was a study area. This is this map actually shows the Hyderabad city, and this is Mehran University. Here is Mehran University. So the Halanaka and the yellow one are the Halanaka connected treatment plants and distribution networks samples, and the green one are the samples from the Vasa and Vasa and its distribution network. These 2 points are from Mehran University treatment plant. So these were the
You have
sample which,
minutes to conclude.
yep. So these are the samples which we have collected: 6 from Halanaka and distribution system, 5 from Vasa treatment plant and distribution system, and 2 from Mehran University treatment plant. And these are the physico-chemical parameters which we have analyzed. All parameters were in permissible limits, and we can see that there was no disinfectant was detected in any samples of collected samples, and only sample from Vasa treatment plant L12 had a higher concentration, high TTS. Otherwise, all samples has, but it was still in not too much. It was in permissible limits, and one sample had the pH greater than 8.5. So after sequencing, these are the results of our sequencing results after processing of the raw reads. So we have got about 7 million 7 million reads in our in total, which after filtration we have about more than 0.47 million reads in our hands, which we further process for taxonomic analysis. So this bar graph actually shows the different phyla which we have observed in our samples. There are about 31 phyla we have observed in the water samples, and the Proteobacteria, Bacteroidetes, Cyanobacteria, Viriocomicrobia, and Actinobacteria were most abundant phyla, bacterial phyla in our samples. And this bar graph shows actually the bacterial genera which we have found in our study. About 430 bacterial genera were identified through 16S amplicon sequencing, and Flavobacteria bacterium was the most abundant genera. However, there were many reads which were unclassified. About 54.5% reads were unclassified, and many genera were below 1% relative abundance. So this PCA plots actually shows the similarity and dissimilarity between the sample. And in pie crust analysis, these are the value for NSTI values for our samples. The value less than 0.15 means that the sample had found its parent sequence very well. Otherwise, when value it increases the value 0.15, it means that the read could not, or the sample could not find very well parents. That's why their predictions can be wrong. So these further results I'm showing, these are on the prediction basis that the genera which we have found that either they had what capabilities they had. So there were many functional capabilities in these identified genera. We can see that many functions they have contributed, and in these functions we can see that human infectious diseases were also the pathways related to the human infectious diseases were also identified, and tuberculosis, Vibrio cholera pathogenic cycle were also identified in these bacteria. Uh, if uh, uh, if the full shotgun metagenomics was carried, might be this was possible, but this is only prediction. So on prediction wise, we have found
Junaid,
that.
please conclude your findings.
Okay, so these are the xenobiotics. So discussion that several studies have been carried out. We have also carried out this research, and we have collected the samples in this spring and winter, and both seasons. In samples are mostly turbid or something contaminated. So there was no residual chlorine was observed. There were many researches which were carried out in which Proteobacteria were found as abundant. Comamendaceae family, which is related to phylum Proteobacteria, is was most dominant, and it has very potential. And the cyanobacteria, which produces cytotoxins, were also present in a sample in Picret, and this is we have found that there were many pathways which were contributing to the human infectious diseases. So 13 samples were analyzed. No free chlorine was detected in the sample. All samples had were in permissible limits. The parameters were in permissible limits, uh, and they have various functional capabilities predicted. And this is acknowledgement. The study was sponsored by Higher Education Commission and USAID. Thank you for your attention.
Thank you very much, Junaid. Is there any question from the participants? Okay, if no question, then I have a question, Junaid. You
Yes.
have shown your, if you go back to your presentation, you have shown the bar charts for the infectious diseases, and I can see the tuberculosis is very high. So all these are the results of the water samples, and the tuberculosis is, I know the Mycobacterium tuberculi. Is it so high?
Yes, ma'am. Actually, these are the prediction based. The reeds which we have found, for example, this reed matches to some genus or something, so it predicts that its parent might have this pathway in his progeny. So might be this would be contributing to tuberculosis or something like those. So these are the prediction based results. But if we go to the whole genome or shotgun metagenomics, these are also, it also predicts the results. It is not confirmatory. So we can say that yes, this tuberculosis may be caused, there are bacteria in our samples which were contributing to this tuberculosis or something like that.
Okay, thank you very much. Thank you.
Okay.
Okay, Bakhtavar Pirzada. So she is going to present her findings on evaluating the antimicrobial activity using Punica ganatum plant species against microbes. Yes, please. And you have 10 minutes.
Yes, ma'am. Evaluating the antimicrobial activity using Punica grantum plant species against surface microbes. Assalamu alaikum, this is Bakhtavar Khair from US Pakistan Center for Advanced Studies in Water under the guidance of Ma'am Aisha Tajammul. Here are the contents: introduction, problem statement, aim of study, methodology, result, conclusion, and reference. Let's start with an introduction. Microbes thriving on a surface such as a floor and bench top are one of the main sources of contamination, and these microbes could easily disperse into the surrounding environment through various routes of entry, which is a serious health and sanitation issue. To overcome such issue, we find plants which contain antimicrobial property against microbes. An antimicrobial is a substance that kill or inhibit the growth of microorganisms such as bacteria, fungi, or protozoa. Nowadays, herbal plants are being presented in market as a substitute and environmental friendly solution against bacteria. Punica grantum. Punica grantum is basically a large and native shrub which belongs to the family Litheraceae. Punica grantum is informed to be used in many diseases conditions in traditional medicine. Various parts of this plant are energetic in management of many diseases. Punica grantum has many prospective effects, including antibacterial, antifungal, and antiviral activities. Antimicrobial properties are noted against oral pathogens. The pomegranate edible and non-edible plants have been reported to be the excellent antimicrobial, antibacterial, as well as antioxidant agent containing rich amount of polyphenolics. Problem statement: to overcome this hygiene and sanitation related issue, various synthetic chemicals are being used. Despite the effectiveness, these chemicals themselves are highly toxic and less biodegradable in the environment and somehow contribute in secondary environmental contamination. Thus, in order to achieve complete cleaning of surface, plants seems a visible option. Aims of the study: this research aim is to explore the possibility of utilizing natural source for producing disinfectant that have potential to eliminate surface bounded microbes. Methods: preparation of a plant material, clinically isolation of surface pathogen, test diffusion method, minimum inhibitory concentration, and kill time method. Isolation for floor microbes. Using Miller-Hunton growth, surface microbes were isolated from kitchen surface using sterilized cotton swab. Different media were prepared to identify the isolated gram-positive and gram-negative bacteria. Bacteria were selected like EMB, isolene methylene blue, was used for E. coli, and MSA, mannitol salt, was used for Cephalo aureus. Disk diffusion method. The antimicrobial activity of a plant were carried out by disk diffusion method. A lawn were prepared on petri plates using test microorganism by spread it with a sterilized cotton swab on the surface of MH hugger. 6 mm of Whatman filter disc was soaked in 100 microliter and 50 microliter of content having different concentration of grind the leaves of Punica grantum. After the discs were placed on a petri dishes using sterilized forceps, and all the petri plates were incubated for 24 hours. Results were recorded in terms of zones of inhibition. These are the 50 microliter of plant contents, and the below one are the zones of 100 microliter of plant content. Minimum inhibitory concentration: two-fold serial dilution of punica were prepared in Miller-Hinton growth in the presence of bacteria growth. The MIC for all bacterial strains were notified with turbidity levels and measured using UV visible spectrophotometer by taking optical density at 600 nanometer. Kill time method: 1 ml of bacteria strain was introduced in 8 ml of MH broth containing 1 ml of plant extract into the falcon tube. OD was recorded by UV spectrophotometer by using 600 nanometer wavelength at 0, 15, 30, 45, 60, 75, and 90 minutes. After each recorded reading, tubes were kept in a shaker at 120 gram, and for the next round to record the reading, the results were noted in terms of absorption. Okay, here is the result for E. coli zone of inhibition. At the 50 microliter concentration, 568 milligram per ml contain maximum zones, and at 100 microliter, the concentration 1135 milligram per ml shows maximum zones. And for Staph aureus at 50 ml, we get maximum zone at 426, and at 100 microliter, we get maximum zones at 1135. For MIC, we see optical density at 0 as the initial reading and the 24 hours as the final reading in the process. We found that the largest difference is 1.0416 at 426 milligram per ml concentration, while at 0.0503 it was 240 milligram per ml at lowest concentration. For Staph aureus, we found the largest difference at 568 milligram per ml, that is 1.7802, and lowest concentration at 240 milligram per ml, that is 0.4747. These are the result of kill time. We found that the Punica grantum has more effect on E. coli than Staph aureus. Conclusion: it is concluded that antimicrobial activity showed that the leaves of Punica grantum have antimicrobial effect on pathogens. The present study also revealed the antimicrobial nature of this plant and it can be used for herbal disinfectant. These are the references. Thank you.
Thank you very much, Bakhtawar. Anyone have a question? You can ask Bakhtawar.
Is my PPT visible?
Yes, it is.
Okay, so my topic is assessment of indoor air quality in terms of microorganisms at USPCAS-W Jamshoro. Let's start.
Full screen kar lijiye.
So introduction: indoor air pollution is caused by various sources such as dust, dirt, and many other sources, and microorganisms also. So different sources are contributing it, like plants, a number of person inside the building, such as many people, like inside a building such a home, like home or any other workplace where the people work, and that contaminations like dust or other microorganisms harm us if we breathe it in. Microbial indoor air pollution involves hundreds of species, bacteria, and fungi that grow indoor when sufficient moisture is available. There are different sources such as peats, humans, plants, soils, direct and indirect of dust. So there are various human health problems such as irritation of eyes, nose, and throat, headache, respiratory disease, heart disease, and so many other harmful diseases. So this study has proposed the aim to determine the level of indoor air microbes, bacteria, and fungi at different locations in USPCAS-W Jamshoro. So I have included the problem statement such as air quality of indoor environment is one of the main factors affecting health, well-being, and productivity of people. People spend 80 to 90% of their time in indoor environment by breathing on average 40 meter cube per day of air. Air quality is affected by presence of microorganisms, which include bacteria, fungi, and viruses. So these are two objectives: assessment of indoor air quality in terms of microbes and fungi at USPCAS-W Jamshoro at various locations, and determine the number of bacterial and fungal colonies at 3 time intervals, such as 30, 60, and 90 minutes. So the methodology, media preparation. First, the media was prepared using a TSB, Tripton soya broth with agar, and the distilled water was utilized, 750 ml of distilled water, in which the TSB 10.5 milligram and 4.5 milligram of agar were added. So the produced mixed media was heated at 120 centigrade for proper mixing, and was heated in autoclave at 121 centigrade for 15 minutes. In the study areas, 6 locations were selected for sampling, such as the reception hall, canteen room, classroom, library, water lab, first floor washroom. In each location, samples were kept at 3 different points, 1 controlled, and 3 open petri dishes were kept at each point simultaneously. Each sampling point was 7 to 8 meter away from one another and 1 meter above the floor level. So the counting technique was used to count the number of colonies. After taking samples from locations in certain time intervals, the samples were kept in incubation for 24 hours. 3 time intervals were selected, 30, 60, and 90 minutes. Bacterial and fungus colonies were counted through the J2 colony counter. So such techniques were applied at all locations. Here is the result taken from the washroom. Number of colonies are shown on petri dishes. So the number of bacterial colonies at different locations, such as reception, canteen, washroom. At washroom, a maximum number of colonies were observed, and while in the water lab library were minimum. And the number of fungal colonies at different locations. So also the number of fungal colonies in a washroom were observed more. So in the samples, the bacterial and fungal colonies were present at the obtained results. At the end, maximum bacterial and fungal colonies were present in samples taken from washroom, reception, and canteen. More colonies were observed in washroom. No colony was present in control samples. Samples which we kept in control, no colony was observed in it. So almost all the locations of department were heavily contaminated with bacteria and fungi. The result is not health efficient. Microbes can cause negative health impacts on anybody who works under such a microbial contaminated area. Thus, the attention must be given to control those environmental factors which favor the growth and multiplication of microbes in indoor environment of department in order to safeguard the health of workers. So these are papers. Thank you. Any question?
Yes, if there is any question from the participants. Okay, Danish, thank you very much for your presenting your findings.
My name is Mavish Nyas, and I'll be presenting my research on biotoxicity potential of lambda-cyhalothrin. Assessment of biotoxicity potential of lambda-cyhalothrin on common carp, and this study was carried out under the supervision of Dr. Imran Hashmi. This is a sequence of my presentation. First of all, NUST, the history, it is primarily a science, engineering, and technology multidisciplinary institute established in 1991, and the main campus is at H12, with disciplines such as engineering, IT, applied biosciences, and management sciences. IESE, the Institute of Environmental Sciences and Engineering, it comes under the School of Civil and Environmental Engineering, established in 1996. Some of the key research areas are environmental toxicology, microbiology, biotechnology, nanotechnology, wastewater, industrial effluents, water quality management, and solid waste management. The pesticide use and contamination, Pakistan, as we all know, is an agrarian country, and significant contribution is from agricultural activities to the GDP. Pesticide imports and use in Pakistan remained unregulated until the Agricultural Pesticide Ordinance, which came in 1971. Extensive use of pesticides in recent times is inevitable in order to accommodate the ever-growing population, and this binds us in a very vicious cycle of essential use of pesticides, whereas also having to deal with the side effects. This graph represents the overall pesticide use in the whole world, and we can clearly see that Pakistan tops the list of the biggest consumers of pesticide, not only worldwide, but specifically in the South Asian region. Pesticide regulation in Pakistan, the legislation pertaining to pesticides is all entailed in the Agricultural Pesticide Ordinance of 1971, whereas at the laboratories and inspectors are nominated at federal as well as provincial levels to monitor the use, import, application, and handling of pesticides. Water reserves and pesticide toxicity, water breeds life, and global freshwater reserves are currently under serious threat. Agrochemicals, pollutants, and toxins, they pave their way to fresh waters through various routes and compromise the water quality. And these toxins are then ingested by the aquatic organisms, particularly the fish. And then all the toxins eventually get transferred to the human community when they consume the fish. In 2017, high contamination in Rawal Lake caused massive killing of fish, which was also reported by Dawn in 2017. These are some of the key studies which were carried out here at IESC, which are also pertaining to the topic that I have worked on. Now comes the introduction. Lambda-cyhalothrin, this is the pesticide that I worked with. It is a synthetic pyrethroid, micro-poisonous to mammals, and it is highly toxic to aquatic organisms. The routes of transport of insecticides and pesticides from the crop fields to the adjacent water streams are via runoff, groundwater, wind drift, atmospheric deposition, or the drainage also. Once water reaches the water body, it contaminates the water body and also disturbs the aquatic ecosystems. Fish are mostly used as environmental monitoring agents because they can accumulate the contaminants directly from the water. Materials and methods. This was the study site, Rawal Lake, which is the major source of water supply for Rawalpindi and Islamabad also. The experimental organism was common carp, the Cyprinus carpio, and they were procured from Punjab hatchery, Rawal town, Islamabad, and they were transported in aerated polythene bags and transferred to the experimental tanks within the laboratory. And then they were acclimatized to the laboratory conditions for about 2 weeks. The experimental tanks were around 3 feet by 2 feet, and they were equipped with air pumps to regulate the supply of oxygen. Fish were fed during the acclimatization period, but once the experimental setup was done, they were not fed. As far as the photo degradation of pesticide, it was done with the help of a UV lamp with the which was of 11 watts and 254 nanometers. The 2 fish tanks each filled with 30 liters of water, and both of them were introduced with 1.25 microgram per liter of lambda-cyhalothrin, which was prepared in a distilled water. And one of the tank was given the UV exposure for about 20 minutes while the other tank was kept as control. And after 20 minutes, the UV lamps were removed and randomly selected fish were introduced in both the tanks. 12 fish were introduced. These are the morphometric parameters of fish. Since the two different experiments were being carried out, one was toxicity assay and the other was UV exposure and pesticide degradation. So 2 different sizes and weight of fish were used, and the details are explained here. For physicochemical parameters, since we were using the tap water, the pH, dissolved oxygen, and electrical conductivity of water was determined initially in order to set the baseline using the standard methodologies. This was the methodology for the whole study. First, for sampling fish, the common carp was sampled and acclimatized, and then its LD50 was determined. And after that, during the experimental phase, they were given the UV exposure to initiate the photolytic degradation. For the experimental design, acute toxicity assay for LD50 common carp for lambda-cyhalothrin experiments were carried out according to the OECD guidelines. And the principle of which was to expose the fish to any test chemical for about 96 hours and observe the die-off rate. The concentrations were selected based on literature, which ranged from 0.25 microgram per liter to 1.5 micrograms per liter. Healthy and even-sized fish were chosen, juvenile fish for toxicity assay. The exposed tanks were then covered with a wooden box in order to avoid the UV component of sunlight to interfere with the photo degradation. And then they were conducted for 96 hours, periodically removing water and calculating the physicochemical parameters such as pH, EC, and DO, the dissolved oxygen. Here are the results and discussion. Q toxicity assay. This figure shows the overall tests done for calculating the LD50. And based on these observations, the experiments were conducted in repeat batches, and the LD50 value was calculated to be 1.25 micrograms per liter. And Habiba and David in 2016 reported LD50 of lambda cyhalothrin for common carp as 1.62, which was very close to the calculated value. These are the physicochemical parameters trend during the 0 to 72 hours. The experiment was supposed to continue to 96 hours, but since it was the initial experiment, fish died before 96 hours, because of which we could not continue until 96 hours. In the first figure, the pH shows an increasing trend, whereas DO and EC both are showing a decreasing trend in both control as well as experimental tanks. The pH of control and experimental tanks revealed increasing trend, and it could be linked to the fact that lambda cyhalothrin might have degraded into its metabolites with different pH, and the reduced EC could be attributed to the fact that amount of organic matter increased within the fish aquaria, reducing the conductivity, and DO decrease could also be explained by the fact that experimental tank was covered with a wooden box, which eliminated the interference of light sources and external oxygen. Here are the conclusions. The LD50 lethal dose of lambda cyhalothrin for common carp was found out to be 1.25 micrograms per liter. pH of lambda cyhalothrin solution exposed to UV light increases with time, and DO and EC both decrease with time. Here are the references and acknowledgements. Thank you.
Thank you very much, Mavish. Are there any questions from the participants?
Assalamualaikum. This is Lareb Shahid. I am MS research student from National University of Sciences and Technology, and I worked under the kind supervision of Dr. Imran Hashmi, and I'm here to present my research work entitled Evaluating the Biotoxicity Potential and Photodegradation of sulfamethoxazole in Water Using Common carp. My presentation layout is as flashed. Starting with a brief introduction. Antibiotics are not only used to treat and prevent human and animal diseases, but they're also used to promote growth of animals in livestock and aquaculture operations. Antibiotics are particularly metabolized in organisms and are excreted as parent compounds or as metabolites. 200,000 of antibiotics have been used annually. This table is showing growth of pharmaceutical industry in Asia. Pharmaceutical industry of Pakistan is increasing at an alarming rate of 11%. A few relevant studies in Pakistan highlighted an alarming situation indicating very high levels of antibiotics. These are the highest detected concentrations in river Ravi. Many of these antibiotics have concentrations in micrograms per liter. Exposure to even such a low concentrations prove detrimental for environment. In Pakistan, there is 16% increase in the use of antibiotics over last 16 years. This is a graph of global antibiotic consumption by country income classification 2000 to 2015, which clearly showing increase in the consumption of antibiotics in the lower middle countries, and especially Pakistan. Possible concerns related to the antibiotics are that they cause microbial resistance, they change indigenous microbial compositions, they disturb to the plant growth, and they cause acute or chronic toxicities on aquatic species, and they cause adverse effects on humans through food chains. But my area of concern was acute or chronic toxicities on the aquatic species. The group of antibiotics from which my antibiotic belong is sulfonamide. Sulfonamides are extensively used against bacterial infections. They also enhance effects against pathogens. Sulfonamide residues detected up to 10,000 nanograms per liter in sewage treatment plants and less than 100 nanograms per liter in water and 998 micrograms per kg in animal tissues. The major concern is sulfonamides are not biodegradable. In sulfonamide, the concerned antibiotic which belongs to this class is sulfamethoxazole, which has a half-life of 10 to 12 hours and has a very low, slow oral absorption. Sulfonamide is considered as an emerging micropollutant. It is used in the human and veterinary medicines, as well as for the treatment of urinary tract infections, for the bronchitis and for prostatitis. Other than that, it causes some of the effects on the ecosystems which lead to the toxicological environmental impacts. Okay, these are some of the physiochemical properties of sulfamethoxazole, and here we have some of the local and multinational suppliers of SMX drug in Pakistan. This was my study area, which was Rawal Lake. This study area was chosen because Rawal Lake is the main source of water for approximately 2 million residents and provide drinking water at the rate of 22 million gallons per day. But the lake water quality is being deteriorated day by day, which results in massive fish deaths in the alarming rates. Some of these were reported in different newspapers. Coming to the literature review. This was some of the literature review which I consulted for my study, and this is the previous work done related to my study in IESC. Okay, these are my objectives: determination of lethal dose of SMX in common carp, degradation of SMX using UV light, and identification of physiochemical changes in water after degradation. Coming to methodology, this is a general layout of my methodology, which includes sampling, acclimatization, then LD50, exposure, identification, and ultimately analysis. For the acclimatization, fish were acquired from a Rawal town fish hatchery, and they were acclimatized for a period of 7 days. And a semi-static tank setup was made for this purpose. Related to the fish specification, the age was approximately 1 year, and the length was 10 to 15 centimeter, and the weight was under 100 grams. These are some of the physicochemical parameters of lake water and experimental tanks. For the determination of LD50, 6 concentrations were chosen: 200, 600, 1000, 1400, and 1600, and 2000 milligrams per liter for the determination of LD50. And for the degradation experiment, a lamp was chosen based on the literature survey, literature studies, and some of the specifications are as flashed. For the dose preparation, SMX tablets were dissolved in the water. Then they were stirred and sonicated for 5 minutes and sonicated, and the suspension which was made and then added to the experimental tanks. This is an experimental setup for which dose was added to the experimental tanks. Then 12 fishes were added in each tank. Then UV exposure was given for 15 minutes. Boxes were covered with the wooden box. Allocates were drawn after 15 minutes, and the samples were stored in the airtight bottles and covered with aluminum foil to avoid any sunlight interaction. And they were stored at 4 degrees for later analysis. Coming to results. Okay, this is a result of LD50. For LD50 analysis, 6 different doses were applied according to OECD guidelines and observed for 96 hours of exposure duration. After 4 days of experimental test, it was observed that no mortality was noticed until 48 hours, but whereas half of the population was killed at 96 hours, and determined LD50 range is 1000 milligrams per liter. The same results were presented by Hong and co-workers in 2014. These are some of the behavioral changes which were observed in the experimental fishes. These were erratic swimming, loss of equilibrium, anxiety behavior, and some more resistance towards the targeted compound. This is a calibration curve of SMX for a UV visible spectrophotometer. For this purpose, a pure sulfamethoxazole in methanol aqueous solution with 0.1 normal sodium hydroxide was used at the wavelength of 200 to 400 nanometer to make the calibration curve. The maximum absorbance was observed at 260 nanometer, and the R square value was 2.9811. Okay, this is a degradation curve of SMX. The smooth degradation curve was observed, and results showed that after photodegradation, concentration of SMX decreases with time. Now coming to the physiochemical results, these are the pH results. pH in the samples, both control and experimental, was slightly higher than the H3 water, but there was not a profound change of pH in allocates drawn after every 24 hours. For the temperature, it fluctuated during the experimental conditions and ranged between 18 to 20 degree, but these fluctuations were mainly because of the overall temperature difference in the environment. For turbidity, immediately after adding the SMX solution, turbidity was more, and gradually it reduced in the sample collected after every 24 hours, and this increased, and this was because of the settling of the undissolved SMX at the bottom of the tank. He water had hardness value greater than 180, so it was a hard water. Hardness decreased in control and experimental groups. For dissolved oxygen, changes in the dissolved oxygen are in accordance with the temperature fluctuations. For chlorine, in control and experimental samples, amount of free and total chlorine decreased with time. Coming to the conclusion, I can conclude that LD50 was determined to be 1000 milligrams per liter for sulfamethoxazole, and the photodegradation was figured out as the most efficient method of SMX removal. Other than that, addition of SMX to the water caused increase in the pH, but decrease in dissolved oxygen, turbidity, electrical conductivity, and free and the total chlorine. These are some of recommendations which are for the future studies related to my work. It can be done that photodegradation pathway of SMX could be found out, and studies to evaluate the gene expression after SMX exposure, then toxicity assessment of a mixture of antibiotics in the fish. At the end, I would like to acknowledge Dr. Imran Hashmi for his kind support, National Universities of Science and Technology, and the MS Research Grant, and HEC for NRPU project for providing grant, and then Environmental Microbiology Research Group, Environmental Toxicology, Environmental Biotechnology, and at last, my friends and family. These were some of the references which I consulted for my study. And thank you for listening.
Thank you, Lare.
Yeah.
Any questions? Okay, thank you, Lare, for your interesting presentation.
Okay, thank you, ma'am.
So thank you very much. So we had 4 presenters, जिसमें से हमारे पास तकरीबन I think just 5 presenters थे who have presented their interesting findings, and I am very grateful to Junaid, Bakhtawar, Lareb, Danish, and Mavish who have presented their findings. And these were very, although the other research topics which have to be presented were quite useful. They were on research, arsenic contamination, treatment, water and sanitation, wastewater quality monitoring, and treatment technologies. But we were very unlucky. We could not have those presenters with us. So, but I hope that their extensive research will have some fruitful impact on addressing the water and environmental issues of the country. And with these comments, I extend my thanks to all the presenters, and I wish all the researchers good luck for their future projects and useful outcome. And thank you very much, Rifa, for giving me this opportunity to moderate this session. And thank you. Thank you all the presenters and all the participants.